Hi, good morning, everyone. Thank you for joining us for today's webinar. My name is Jessica Parker, and I'll be moderating the event on behalf of Florida Society for Clinical Oncology, Gilda's Club of South Florida, and the CLL Society. Today, we are presenting three perspectives on CAR T-cell therapy from Dr. Frederick Locke, Program Co-Leader, Immunology, Blood and Marrow Transplant, and Cellular Immunotherapy at Moffitt Cancer Center in Tampa, who will provide an overview of CAR T and the therapy process. Also from Dr. Krishna Komanduri, Medical Director, Adult Stem Cell Transplant Program, Sylvester Comprehensive Cancer Center in Miami, who will speak to issues related to access and coverage. And finally, from Dr. Brian Kaufman, Co-Founder, Chief Medical Officer, and Executive Vice President of the CLL Society, who will give us the patient perspective from his own journey through the cancer diagnosis and treatment process. Just a little housekeeping before we get started. If you have questions during the presentation, please type them into your question box in the GoToWebinar panel, and those will be seen by the moderator only. We will have time for questions at the end of the presentations. Without further ado, I will turn the time over to Dr. Locke.
Thank you so much, and thank you for organizing this today. And I'm pleased to start off by giving you an overview of CAR T-cell therapy. And I'm coming to you from Moffitt Cancer Center, where I'm a physician scientist, and clinical researcher. So what are CAR T-cells? Well, they're an autologous adoptive cell immunotherapy, meaning we take the cells out of the patient themselves, at least in their current form of CAR T cells, and we give them back to the patient to treat their cancer. These are T cells that are removed from the peripheral blood, and they're engineered using a gene, this is a gene therapy, to express something called a chimeric antigen receptor. That's what CAR stands for, chimeric antigen receptor. And these T cells are now reprogrammed to recognize cancer cell targets. They essentially have a little GPS navigation on the surface of their cell that tells them to go against a single target. Normally, we have these T cells circulating throughout our body, in our peripheral blood and in our lymph nodes, and each T cell could recognize a different target. Some T cells could recognize the flu, others could recognize bacteria. And in the case of CAR T cell therapy, we've reprogrammed all of those T cells from the patient against a single target. And the target CD19 is the one that now has FDA-approved CAR therapy. This is a picture that can kind of help you visualize a little bit closer of how the CAR T cell interacts with the tumor cell. This big round circle here is a B cell tumor that expresses a protein called CD19. And the T cell, or in this case, the CAR T cell, can recognize those cells because it now expresses an artificial protein right here that spans the cell surface membrane of the T cell. And it contains an extracellular binding domain that recognizes CD19 on the surface of this cancerous cell. And then intracellularly, it has some signaling domains that activate the T cell for a fight, basically telling it to attack this target when it finds this CD19. And this is really building upon the natural way that a T cell is activated when it recognizes something, and it has some co-stimulatory signals that are required in order to make it activated. In this case, they're built into the CAR molecule itself. Now, CD19, as I mentioned, is a protein on the surface of B cells. These are different immune system cells, and it really is the ideal CAR T cell therapy target. Why is that? Well, because CD19 is on the surface of all B cells, and that includes precursor B cells and mature B cells, from little baby B cells to more mature B cells, and all the corresponding cancers that can arise from these different B cells, including acute lymphoblastic leukemia, marginal zone and mantle cell lymphoma, chronic lymphocytic leukemia, diffuse large B cell lymphoma, and follicular lymphoma. And in addition, we can target normal B cells, and we know that we can survive without the normal B cells in the circulation. So as a byproduct, normal B cells may be destroyed by CAR T-cell therapy against CD19. So the first clinical responses with CD19 targeted CAR T-cell therapy was seen in lymphoma, CLL, and ALL, and was described about 8 years ago from several groups that were studying CAR T-cell therapy in single-center trials. This includes at the NCI, University of Pennsylvania. There's also work done at Memorial Sloan Kettering and Fred Hutchinson Cancer Research Center, and among others, Baylor. And these reports demonstrated that patients who had a tumor, whether it be lymphoma here on the left, or CLL in the middle, this is the bone marrow section, or acute lymphocytic leukemia, patients could have remission through a single infusion of the CD19-directed CAR T-cell therapies. This slide shows you what sort of the process is for a patient going through CAR T-cell therapy and how we get through the journey, and you may hear more about that later. But essentially, when we have a patient, the first question is, could they benefit from this CAR T-cell therapy? And if so, we undergo vital organ testing, heart, lungs, kidney, liver, and make sure that the stresses of the therapy could be tolerated safely or as much as possible. And then the patient undergoes leukapheresis, which is a procedure that's probably most familiar to describe it as sort of like a dialysis machine, where the patient's blood runs through this machine and the white blood cells are collected out. And then those cells are shipped to a central facility where they are manufactured and turned into CAR T cells. And they return back in about 3 or 4 weeks. We give the patient some conditioning chemotherapy, not so much to get rid of the lymphoma or the leukemia, but to make space and get rid of the existing normal immune cells that are there, so that when we infuse the CAR T cells in, they can proliferate and expand, and it becomes a living drug. And in the blood where they're infused, they know where to go because of this CAR that's been placed into them and is on their surface. Patients generally stay in the hospital for the infusion, and for 5 to 21 days after, we follow for certain specific toxicities, which I'll mention in a moment. There are some CAR T-cell therapies that have been administered as an outpatient, and that may be in the future. We'll give more of these as an outpatient. And generally, our first tumor assessment is at day 30 after the CAR T-cell therapy. I'm going to speak briefly about the ZUMA-1 trial. This is a multi-center trial in patients who had refractory aggressive non-Hodgkin's lymphoma. Just as background, diffuse large B-cell lymphoma is the most common form of non-Hodgkin's lymphoma, 26,000 cases per year in the U.S., 10,000 deaths in the United States each year from non-Hodgkin's lymphoma. And for patients who are refractory, meaning they did not respond to their last line of chemotherapy, they have a chance of responding to their next line of about 1 in 4, and a chance of having a complete response in less than 1 out of 10 patients. And so really, this is a poor prognosis for these patients with refractory diffuse large B-cell lymphoma. And these are the patients that we enrolled onto the ZUMA-1 multi-center trial, and we saw a response rate of 82% with a single infusion of CAR T-cell therapy, and a complete response rate in about half the patients, and durable responses beyond a year, importantly, in about 40% of the patients. This slide shows you a photomicrograph or a PET scan, PET CT scan overlay of a patient at Moffitt who had multiple prior lines of therapy. And this gentleman, you can see all the black areas involved with lymphoma. And after a single infusion of CAR T-cell therapy, axicabtagene ciloleucel is the name of the CAR T-cell therapy. At 3 months, complete disappearance, and you can just see some uptake in the kidneys here from the PET scan tracer. And so this trial led to FDA approval of this particular therapy called axicabtagene ciloleucel, again, CD19-directed CAR T-cell therapy for adults who have relapsed or refractory large B-cell lymphoma who've had 2 or more lines of therapy. This isn't the only trial that's led to FDA approval of CAR T-cell therapy. A month or 2 before that, a different CAR T-cell therapy was also FDA approved. That therapy is called tisagenlecleucel, and this was FDA approved for pediatric patients and adolescent young adults up to age 25 with acute lymphocytic leukemia. And that same therapy, tisagenlecleucel, was also FDA approved based upon a clinical trial called Juliet for adult patients with refractory lymphoma. So we now have two different CAR T-cell therapies that are FDA approved with indications including ALL and diffuse large B-cell lymphoma. The therapy doesn't, it can work and it can lead to long-term durable remissions in these patients, but it's not without some risk. There are toxicities that can happen with CAR T-cell therapy. The two main categories are called cytokine release syndrome and neurologic toxicities. And you can imagine that if you were to get the flu, you could have high fevers and low blood pressure. Similarly, if you reprogram the T cells of the immune system against a single target and they go and start to attack it and kill the cancer, you can have similar effects from the immune system. And patients may have high fever for days at a time, in some cases low blood pressure or problems oxygenating. And then separately, there's a class of toxicities that we see sometimes that can be that are associated with the central nervous system, things like confusion, word finding difficulties, and in some cases we see seizures. Both these categories of toxicities are generally reversible. In almost all patients who receive CAR T-cell therapy that have these symptoms, they resolve within the first month after therapy. This just gives you a timeline of the discovery of CAR T-cell therapy. And although the clinical trials have happened relatively rapidly, this therapy did not just happen overnight. And scientists have been studying the concept for about 30 years, just highlighting the importance of investment in research and continuing to improve upon the therapies that we have. The American Society of Clinical Oncology named CAR T-cell immunotherapy the advance of the year in January of 2018, in part from some of the results I've shared with you today. CAR T-cell therapy is also being tested against solid tumors. This slide shows you a patient with glioblastoma. You can see in the red circles here tumor that's growing in near the brain or inside the brain, and then also in the spine. And you can see after infusion into the central nervous system of a CAR T-cell therapy against a different target than CD19 that there's regression of the lesion. And so there will be more targets and more CAR T-cell therapies being tested as we go forward, but CAR T is not the only cell therapy in development. You can see here, as of March 2018, there were over 700 trials testing cellular therapies, and over half of those were CAR T-cell therapies, but other treatments include something called T-cell receptor or TCR therapy, which is a close sibling to CAR T-cell therapy in that it's a gene therapy, but it uses slightly different mechanisms of the T-cell to activate. And then other things like tumor-infiltrating lymphocytes, where the T-cells that are within the tumor are taken out and grown up in the lab and infused back into the patient. All these cellular immunotherapies are being tested in multiple centers, not just in Florida and not just in the United States, but around the world. And in fact, you can see here the three sort of hubs of development of cell therapies: the United States, Europe, and then China. Again, highlighting the importance of investment in clinical research so that we can make progress and improve outcomes. And so finally, CAR T-cell therapy is effective, and it really provides hope for patients who are without other hope. There are many unanswered questions. These are just a few of them. Can we make CAR T-cell therapy safer and more effective? Will CAR T be effective as an earlier line of therapy? These were patients who have had multiple lines of chemotherapy. Could it be given earlier, safely and effectively, and maybe improve on the outcomes that we see with chemotherapy? Can we develop an off-the-shelf CAR T-cell? It's many smart people and companies are looking at this. It's very complex because of the immune system is complex. And so there's a lot of science that has to be worked out. And then finally, when will CAR T-cell therapies be effective for solid tumors? And with that, I'm going to pass it over to Krishna Kamanduri to continue the webinar. Thank you.
Great, and while I'm waiting for the slides to hand over, I just want to thank the organizers for letting me be part of this series and to join my friend and colleague, Dr. Locke and Dr. Kaufman, to talk about, again, varying aspects of this process. So I hope you have my screen up. And so I'm going to actually try to complement what Dr. Locke had talked to you about, which is really the overview scientifically and some of the really exciting clinical trial results. And I'm going to show you just a little bit of that, but really try and talk about some of the global challenges with respect to implementation and especially the finances, because these are unprecedented therapies in terms of their success. They're very, very important here in Florida. I will add Dr. Locke at Moffitt, his center, and our center here at the Sylvester Cancer Center in Miami were two of the four centers that contributed most patients to the pivotal trial that led to the FDA approval of Yescarta or Axicell, as Dr. Locke just showed you. And we have also in the phase of commercial deployment of these therapies, as they now reach patients outside of clinical trials, have treated among the largest numbers of patients in the country. And that's not terribly surprising given the number of patients in Florida who have cancers and also the higher age of Floridians compared to the national population, because these diseases, especially diffuse large B-cell lymphoma, are diseases that increase in incidence with age, as with CLL, as will be addressed by Dr. Kaufman. So I want to, just give you a very high-level overview of why this is so exciting, as Dr. Locke has suggested. So this is a painting from France that actually showed the guy in the middle, who's Dr. Alfred Velpeau, who described this condition of acute leukemia, which is, again, one of the diseases that expresses CD19, which is the target that can be treated with these first approved CAR T-cell therapies. And he described the case of a florist who came in with a white gruel-like consistency of his blood and a massive enlargement of his spleen. And this patient, typical of patients throughout history, unfortunately, died within the initial diagnosis. So the diagnosis was made at that point, and we recognized that malignant cancer cells could either take over the bone marrow or the lymph nodes or both, but there was very little that we can do. And this next slide highlights the fact that really between 1825 and 1950, there were really only about 1,000 publications about leukemia in those 125 years, and really no effective therapies at all. Between 1950 and the year 2000, however, there was really an explosion of knowledge, and first in terms of diagnosis and understanding the biology of leukemias and lymphomas, but also then the advent of new approaches that could, for the first time, start to improve survivals. And in the interest of time, I'm not showing you those data, but in the 1960s, combinations of chemotherapy drugs started to put a dent in survival, that is, put a dent in mortality and improve survival. And at the beginning of the 1960s, almost nobody, pediatric or adult, lived at all with any of these diseases of leukemia. But by the late 1960s, a subset of individuals, 20 to 50%, depending on the specific diagnosis, were cured with combinations of chemotherapy. We also started to do stem cell transplants. Dr. Locke and I are both stem cell transplant physicians, and we learned that by giving chemotherapy and giving an immune system from a donor, we could also cure patients with stem cell transplants. And indeed, since then, we've recognized that the way that those donor stem cell transplants cure patients is through the donor immune system, and specifically T cells. So we recognize that T cells, which normally keep most of us free of viral infections and bacterial infections and infections from fungal organisms that we encounter daily, can also have potent effects in treating cancer cells. And as Dr. Locke explained, cells can be engineered outside the body now to redirect them against cancer cells in this way. So this basically shows this progression from 1825 to the 1960s to 1970s, where we first started to have curative therapies, and then in the 1990s, really recognizing that T cells were critical for transplant cures, leading to the advances that Dr. Locke described, roughly 10 to 15 years ago, the early advances that have now been translated to the dramatic clinical trial results that he showed you. This is Emily Whitehead, who was the first child treated with CAR T cells for pediatric acute leukemia. Emily was literally on her deathbed and ran out of therapeutic options when she was treated by the group at Children's Hospital of Pennsylvania with CAR T cells that, like the therapies that we're talking to you about today with lymphoma, were directed against CD19 and put her into a remission that has been lasting. This is a lovely picture of Emily in a picture that she likes to repeat every year, showing that she was 5 years cancer-free. And Emily, among other things, has started a cancer foundation. And I am stuck here trying to get the next slide. There we go. And this is a picture of Emily visiting the last resident of the White House, and the excuse note that President Obama wrote from her saying, "Please excuse Emily from school. She was with me," suggesting how remarkable the comebacks can be from advanced disease to really normal lives without other therapies other than immune cells. So we are really here at the point where, again, as Dr. Locke pointed out, in late 2017, just a little over a year ago today, we had the approval of the first two engineered T cell therapies, so gene-modified expanded T cells. And what I'd like to show you next is really an example of what this can look like. And Dr. Locke suggested that this is a living drug. And what I'm going to show you is a little video from a friend and colleague, Stan Riddell, who's been a pioneer in immunotherapy at the Fred Hutchinson Cancer Center. So what you see here are the elongated cells that look kind of triangular and longer, are actually brain cancer cells in a dish. And the little cells that look more like stars and are kind of more shimmery here are T cells that you're going to see as I roll this video are going to start attacking cells, and then they're going to keep moving and multiplying to attack the cells. And you can see that in the beginning, there are many of these solid tumor cells, these brain cancer cells in the dish. But as we roll this clip that shortens time over an hour or two here, we see that the cancer cells are being destroyed, and the T cells are in fact dividing. So there are actually more of them at the end of this loop than at the beginning. And this is, although we can't illustrate this so nicely in the clinical setting, this is exactly what's happening inside the bodies. That, as Dr. Locke pointed out, that this is really a living drug that's now resident and ready to control the cancer if it should grow again, which is quite different than how we treat patients with historical chemotherapy. So this is an overview of the manufacturing process. This is a slide from David Porter at Penn, who's been a pioneer in this area, and using a term that Dr. Carl June, also at the University of Pennsylvania, has termed creating serial killer T cells in the body. Again, as Dr. Locke pointed out, cells are collected, white blood cells are collected by this process called leukapheresis. They're genetically modified and expanded dramatically outside the body, and then they're basically returned after quality and release testing. Patients are given a bit of chemotherapy, and these modified T cells are infused into the body. And this process typically, again, takes anywhere from a couple of weeks to a month or so for the manufacturing process. So there's really a patient journey. So there's this apheresis process. The cells are actually typically shipped to a central facility. And during that time, the manufacturing period, the patients return home. The patients may be given chemotherapy because of the aggressiveness of their disease. And then they get lymphodepletion or a little bit of chemotherapy that's not very intensive, but prepares their body for the T cell infusion, and the T cell infusion is given. And again, the two primary targets for the approved therapies are acute lymphoblastic leukemia and non-Hodgkin's lymphoma, both of which share this target called CD19, as explained nicely by Dr. Locke. I just want to show you one of our slides from the first patient that we treated here at Sylvester. This is a woman who, as Dr. Locke pointed out, these black spots on this PET imaging are abnormal areas. This is a massive spleen that's replaced by lymphoma, and these are actually masses of lymph nodes that are around the aorta, the major vessel coming out of the heart, and could have led to a fatal complication. 2 months later, the patient actually was in a complete remission. Unfortunately, that complete remission, while it was sustained in April, led to progression of the disease again in July. She was actually retreated with multiple therapies, and unfortunately is not free of disease, but is still alive now 3 years following her initial diagnosis and treatment with CAR T cells. So again, not a perfect story, and I think highlights that, although we have dramatic responses in patients like this who we would have expected had less than a 5% chance of any meaningful response, we do also have challenges and relapses that still continue to occur. So I'm going to move forward here. So our pipeline, as Dr. Locke pointed out, there are over 400 CAR T clinical trials that are open now across the world. More than 85% of the products in the developmental pipeline have been developed to treat various types of cancers, both blood cancers, hematologic malignancies, as well as solid tumors. These include the therapies for which there are approved treatments right now: acute lymphoblastic leukemia, non-Hodgkin's lymphoma. Probably next to be approved will be a treatment for multiple myeloma and chronic lymphocytic leukemia, although we don't have approvals, as we actually know that some of the first patients treated with these really dramatic transformative approaches were patients with CLL who can also express the same CD19 antigen, but there are also trials that have at least had some early successes in pancreatic cancer and glioblastoma, but we're much further away from demonstrating the kind of success that we've shown in the blood cancer side. And again, this is just showing that the current approved therapies are the tip of the iceberg. This is what was approved in 2019, and these are some of the many different antigens and the different types of cancer that are being targeted in these clinical trials and are in the pipelines of many biotechnology companies right now. So the complexity of CAR T-cell patients, these are not easy therapies to deliver and really should be delivered in academic centers like Moffitt, like Sylvester, and other experienced centers that have an experience with cellular therapy, often are blood and bone marrow transplant centers. So these patients have advanced, very frequently very refractory leukemia, lymphoma. The patients may require additional chemotherapy and often have very compromised functional status because of their aggressive disease. I'm sorry, I'm losing this here. Up to 20% of patients, I don't know why it's flipping back and forth, suffer disease progression between the time that the patient is collected and the CAR T-cell product is ready for infusion. And as Dr. Locke pointed out, cytokine release syndrome or this inflammatory syndrome or neurologic toxicities happen relatively commonly in 20 to 50%, depending on the severity that you're examining. You're looking at 50% of these patients will require ICU management at times. There have been fatalities. We ourselves have seen fatalities, and this requires a multi-departmental infrastructure and crosstalk, and the clinical setting is very important. So there are unanswered questions. Patients who present with DLBCL, for example, lymphoma, are eligible for CAR T-cells. Some can have no response or treatment-associated death, despite the phenomenal responses of around 40% at a year and a half or 2 years. That means that 60% of patients are not cured with these therapies. There's a subset with sustained response to CAR T-cell alone, and then there are some who need another treatment like a donor transplant to cure them. And I think one of the health economic questions is the value of CAR T-cell therapy the same across all three groups, and should it be the same? And that's quite important because the two therapies for non-Hodgkin's lymphoma by Novartis and by Kite Gilead have been priced at a remarkable $373,000 for one infusion of cells. The price for acute leukemia is actually higher than that, around $475,000. So these are unprecedented therapies in terms of their likelihood of inducing response, but also in terms of their cost to the system. And this really raises a number of health economic questions and has placed stressors on systems, both in private payment, but especially public payment, including Medicare, as I'm going to discuss. So what does this mean in terms of issues? Well, the American Society of Blood and Marrow Transplantation, and I was privileged to serve a term as the American Society for BMT's president last year, has recognized that societies like ours can promote science, state-of-the-art clinical medicine, and evidence-based standards, that we need a regulatory framework like we have for allogeneic or donor transplantation, and that we need registries so that people can see how individual centers are doing and how we're doing collectively, so that we can have cooperative research and transparent outcomes reporting. Because these therapies are unprecedented, we don't want people to be doing them with results that are not visible and with outcomes that are not clear to both patients and to other scientists and physicians. So there are a number of advocacy organizations, Be the Match, that runs donor transplant registries, the International Blood and Marrow Transplant Registry, the Foundation for Accreditation of Cell Therapies, which accredits cell therapy programs, including stem cell transplant programs, and these T-cell therapy programs and professional societies that are really working together to address these challenges. So right now, one of the main issues is that providers bear the cost of CAR T-cells. For Medicare patients, and remember the median age of diagnosis for patients with lymphoma is above 65, and for CLL, it's often in the 70s. The current Medicare inpatient system is really woefully inadequate. Right now, that cost of the product, that $373,000 cost for lymphoma, which is really completely out of our control as academic scientists, it's set by pharma companies, is not reimbursed by Medicare, and I'm going to explain how that reimbursement is handled right now. And so therefore, the entire cost of providing this transformative therapy right now is being partially shared by providers when patients have, for example, something like Medicare. This is an unsustainable situation, and some centers are actually doing this at great loss, and other centers are not actually addressing or treating care of patients who have Medicare because of the significant shortfalls, and I'm actually going to describe what this is like. So most hospitals in the country are what we call PPS hospitals, all but a small number of academic cancer centers, and Moffitt and Sylvester happen to be among the cancer centers that are actually not under this PPS system, but the vast majority of cancer centers where patients are treated are part of this PPS system. For patients who get treated with lymphoma, there is what's called a DRG, or a diagnosis group, that's assigned, and the national unadjusted payment rate is about $39,000. Now, and that's for both the infusion of cells and the episode of care. So recently, there's been a system due to lobbying by both physicians as well as companies that are developing these therapies. There is a new process for recently developed technologies, which is something called a new technology add-on payment that centers can receive. But, and it's more complicated to explain how they would get this payment, but the maximum NTAP dollars a hospital can receive is $186,500, which is basically half the cost of the product. So you can imagine if you are caring for a patient, you have to pay $373,000 just to get the cells from the manufacturer, and now you have to hospitalize the patient for what may be a week, might be 2 or 3 weeks, and you're getting paid a total of $186,000 plus $39,000. The only thing you can do is actually lose money. These are certain absolute guaranteed losses that are really unsustainable. So there is an outlier payment, but this still results in being paid far less than what it costs just to purchase the drug. And our calculations, depending on how people price the various things that they include in their system, suggest that most people will lose at least $50,000 to $100,000 before they take care of the patient at all right now under Medicare reimbursement guidelines. Now, so what does this mean for hospitals like Dr. Locke's hospital, like Sylvester, which are PPS exempt hospitals, which gives us certain advantages as academic cancer centers in the way that we treat most cancer patients? Well, it turns out that Medicare reimbursement methodologies for our hospitals is actually different, but does not adequately account for the cost of innovative therapies like CAR T-cells. And the inpatient rule, which determines how Medicare pays us and the more typical PPS centers, did not provide any relief for these institutions. And importantly, they are not eligible for that NTAP or outlier reimbursement. So our risks are actually very, very high and not lower than the risks for other hospitals who want to provide these technologies to the patients who are literally lining up at the door for many of us. Medicare does have a better outpatient payment process. And for a patient who's treated in the outpatient setting, they actually pay us the average sale price of the drug. So they will pay us the cost of that acquisition plus a 6% if the product is infused in the outpatient setting. But for all of the patients who are treated on the ZUMA-1 study that Dr. Locke and our patients participated in, all of those patients were treated in the inpatient setting because complications are frequent and often happen very early. And we don't think that it's safe to administer that therapy in the outpatient setting for the vast majority of individuals. Even for the Juliet study, which looked at the competing therapy that's on the market, the majority of patients were actually treated in the inpatient setting. Some were treated in the outpatient setting, but a significant number of those were admitted to the hospital. So we have a situation where there is far better reimbursement now for outpatient care, but not in the inpatient setting, which could force the hand of some providers to try to provide this care to patients by doing care in a setting that we don't think is the optimal setting, given the toxicities that we talked about. So providers that are certified may have to slow the rollout of these programs. Some providers that are already certified by the manufacturers, that is, vetted to be able to deliver this cost in these therapies in a safe way with high and expected good outcomes, have chosen not to provide these care. There's a pressure to shift some of the therapy to the outpatient setting to recover the product acquisition cost. We feel very strongly that the site of care should be cost agnostic, and some providers are focusing only on accrual into clinical trials because they don't want to treat patients with the commercial product. The losses become dramatic and are concentrated in those centers that are willing to do this, which really leads to a lack of sustainability and some very tough choices. So we are proposing as a society, we've actually spent time with, as a society with Seema Verma, who's the administrator for Medicare and others, and are proposing basically a cost to charge ratio so that we don't get a fraction of those charges, but rather the whole cost of the product directly reimbursed to us rather than a small fraction as I talked about. So our organizations believe, and this includes the American Society of Hematology and the American Society of Blood and Marrow Transplantation. These slides were actually taken from slides that were presented by Navneet Majal, who is going to be the president-elect for the American Society of Blood and Marrow Transplant, to Senate committee staffers in Capitol Hill yesterday, so they're really hot off the presses for that congressional presentation. But what we told them is our organizations believe that this interim solution could address the large financial losses associated with the provision of CAR T cells now and help to preserve patient access to care while more durable solutions are being vetted. And we think that then they can use this as a bridge to basically defining a better, more long-term solution. So our congressional ask right now is to ask the CMS to adopt the intermediate technical coding changes outlined in our letter submitted to the Administrator Verma's office, and these are actually on the website, asbmt.org. We've urged CMS to include and adopt this charge ratio and working with stakeholders in CMS to ensure that new payment models don't penalize providers for delivering medically appropriate care, and then working basically to provide a system. Now, the good thing is that private payers, traditional insurers are typically allowing us not to get any profit on that huge cost of the $373,000 cost, but at least paying for that so that we're not having any losses, and then paying us appropriately to take care of these very complicated patients. So this is really a problem with private payers, not with private payers, but with public payers, both Medicaid and Medicare. Some states have dealt with this in Medicaid legislation, but Florida has not comprehensively dealt with this yet. So I would just say that there are really a number of issues. We actually want to get to the point where financial models will actually pay perhaps for outcomes. And in fact, I'm part of an MIT think tank called the Focus, which is financing for curative therapies in the U.S. There's a great story on NPR that's available at npr.org this morning that was interviewed one of the faculty members in economics at MIT who's leading this collaborative group that talks about these financial challenges and how even private markets are trying to deal with these cost-based issues. So I'm going to leave you with, I think, more positive, hope. We went from really no effective therapies for these diseases for really 150 years after we started to diagnose them, to the chemotherapy era that started in the 1960s and started to cure patients, to the stem cell transplant era, which we realized was really a combination of chemotherapy and immunotherapy. And I think we are certain, individuals like Dr. Locke and myself, who are physician scientists and advocates, like Dr. Kaufman, can tell you that we are going to have in our future increasingly effective immunotherapies. But in the meantime, we're going to have to figure out how to deal with this as a society, how to create patient access and how to solve these financial problems, especially with public payments, so that we can sustainably and thoughtfully create the therapies for tomorrow. So I'm going to turn the microphone over to Dr. Kaufman now, and again, thank you for your time.
Hi, Dr. Brian Kaufman here. I want to thank the Florida ASCO and Gilda's Club for this opportunity, and to Dr. Locke and Dr. Comanderi for their introduction to this. And I'm going to have quite a different perspective, and that's because I am not a hematologist. I'm not a transplanter. I'm a family doctor, but I'm also a CAR T patient, and that's what I'm going to be talking about here. And I'm going to try to have more of an upbeat perspective on this and kind of more of a patient perspective on it. And I'm going to kind of skip over the science, which I think has been mostly presented already. So I'm going to tell this story from a different perspective here. I'm going to tell this story from the perspective, a little bit of the T cell, and also from my own perspective and from my caregiver's perspective. I'm the co-founder and the chief medical officer of Yale Society, which is a not-for-profit, which is dedicated to the unmet needs of the CLL or chronic lymphocytic leukemia community, where CAR T therapy is still an experimental therapy and not FDA approved yet for treatment. I wanted some of the stuff that Dr. Comanderi talked about laid heavily into my decision. And in a lot of leukemias, lymphomas, there's multiple pathways patients go down. And this is particularly true in CLL, where we often, as a patient, have to make a decision that can be life-changing with conflicting advice and imperfect knowledge, and certainly making a decision to have CAR T therapy, where it's still experimental in CLL, fit this category. So why did I decide to move ahead with CAR T therapy when there were still potentially other options for me? Well, one of the reasons that I was moved, and not the only reason, but one of the reasons, was one that Dr. Comanderi talked about, which was I could get into a clinical trial. There was a window open for me to get into a clinical trial with a different CAR T product, and this is out of the Seattle Cancer Care Alliance, the Hutch, which had, in early data, in just really a score of patients, had shown strong results. And I could get into this, and I could get my insurance to pay for it. But I'm over 65 and would be switching to Medicare. And I was very uncertain whether CAR T would be an option for me when my insurance was Medicare and if I needed it a year or two down the line. That was one factor. The other factor was I've had multiple treatments for a very difficult to treat, refractory, aggressive CLL with multiple complications. And CAR T offered a whiff of a hope that these responses could be extremely durable and, potentially, and I'm hesitant to use the word, but potentially curative. We have people that are now over 7 years out with CLL who had CAR T therapy at UPenn who still have no evidence of disease and persistent CAR T cells. This was certainly appealing to me, and the early data was very exciting. So the way I tell the story on the website is with a cartoon, and we introduce a lymphocyte who gets transformed and the T lymphocyte, and I've skipped all those slides. But I want to talk a little bit about the process that you've heard already, and I'm going to kind of skip over most of this that's covered, but just talk about it a little bit from the patient's perspective. And the first is you have to be screened and make sure that you're stable and well enough. And this is not a trivial matter. In CLL, the average age at diagnosis is 72, and most people aren't getting CAR T at 72, getting CAR T years after diagnosis when they failed other therapies. So that's not a trivial matter to see what other comorbidities and other issues these patients have. Once you're screened and you're psychologically and medically okay to go ahead with it, you heard about the apheresis process. I'm going to show you a slide of that like. And then there's the waiting. And one of the advantages of my therapy at the Hutch was the manufacturing is done right there, so there's no chipping away, and the turnaround time was quicker. In CLL, the disease doesn't tend to progress as rapidly as with other cancers, so that wait time is less critical, and shortening that wait time is one of the important pieces that have got to be put together. The lymphodepleting chemotherapy was to prevent my body from rejecting the CAR T. That was done in the way they do it at the Hutch was on an outpatient basis. It was really quite gentle. It was not to knock down the CLL, but it was more to allow space for the CAR T cells to come in. I have to tell you that getting the CAR Ts, and I'm giving the patient's perspective, and I've also had a failed allogeneic or unrelated donor bone marrow transplant 10 years ago. So I've been through, this is my second cellular therapy. I have, like I say, quite an aggressive form of CLL. It's somewhat anticlimactic. You just get this sort of bag of cells. It takes maybe 20 minutes, and nothing happens. I mean, nothing happens at the time. And again, and at the Hutch, they did this on an outpatient basis, but you're right there. And my wife, my caregiver, had this huge list of, if this happens, you call the hospital. If this happens, you call 911. There was all these lists on the fridge and everywhere about what we had to do. And then you wait. And this is, again, I want to just point this out a little bit. It's very strange because you're waiting and you're hoping that you're going to get a little bit sick. So this is all on an outpatient basis, and I have strict instructions not to be more than 20 minutes away from the hospital. But it's an outpatient basis, and you're hoping that you have a fever, or you get achy, or you get sweaty, that something starts to happen, because that suggests CAR T are beginning their job as serial killers. I call it kind of a killing frenzy. And you want to get sick, and you want to get a little bit of the cytokine release syndrome. And although the hematologist here can tell that there's not always a one-to-one correlation between having this cytokine release and the response, most people who have a good response have at least some cytokine release. My response was also complicated by some joint inflammation, and I'm going to talk about the restaging I had at 1 month and again at 6 months. So here's a picture of me, and this is in the background. You can see the leukapheresis machine, and that's the raw material that they pulled off my T cells and they made the CAR Ts from, and they pick it up and just kind of walk it over to the manufacturing lab. It wasn't a terrible process, except if you look carefully, you see I have a needle in both arms, one the blood coming in, one the blood going out. And then, again, I'm skipping all the steps, but the CAR T cells are transformed in the way you've heard about. This is our cartoon. I do want to spend a minute on the caregiver, because with the CAR T therapy, it's critical that you have someone who can look after you. And here's my wife. And again, you can see this is an outpatient setting at the Hutch where I'm being closely monitored. But I couldn't do this without my wife. My adult daughters came up. It's very important to have that kind of support with you. And again, I skipped through some of the slides, but you've heard this kind of stuff. And what happens is, after this sort of killing frenzy and those cells that the CAR T has its foot on there are my cancerous B cells. They were destroyed when they looked at my bone marrow at 1 month. It looked like the description was there was these whole areas of debris fields and ghost cells where there used to be lymphoid aggregates of the cancer cells. It almost looked like it had been bombed out. That there was no cancer cells to be found. And there's different ways that they can test for this, but one way is they looked at a molecular marker for the cancer, and then they looked for it again that when my bone marrow was full of cancer, about 60% cancer, and then they looked at it again after, and they couldn't find any of it. And that test goes down to 1 in 100,000, or some say even 1 in a million cells. So there was absolutely no evidence of cancer in my blood, in my bone marrow, and all my lymph nodes, which were quite enlarged, had all shrunk back to normal again. This is how, if you want to see the whole comic book and stuff, which I think is kind of a fun way to explain this is an access to that. So if you do take a look in the mirror here at the doctor here who's telling you that he thinks you'll find him one of the most empathetic doctors around, just glance in the mirror, because I think when you're a physician and you go through this, you have an added responsibility to share what you've learned and try to explain it in patient-friendly terms, and that's what I've tried to do by setting up for the not-for-profit. And this is just a list of some of the things that we offer to patients to try to explain. This is our website. And I, too, had a meeting with CMS, but I went as a patient, not as a physician, with a couple other CAR T patients from around the country. And what I want to point out is for a lot of people, like myself included, CAR T may be the only option that people have. And the success rates, especially in CLL, are extremely high now. And there are very significant adverse events, as you've heard about, but almost always they're manageable. And the treatment-related mortality, while it's there, is quite small. And again, I'm not a transplant specialist, but it seems to me these numbers are smaller than what we see with bone marrow transplants. And it offers the potential for these very durable responses that are beginning to sniff like, these could be lifelong responses for some patients. So these are some of the things I talked about in my meeting with CMS. And as you saw in these pictures, we're not at the beginning of CAR T therapy. This is a third or maybe even later generation of CAR T. But this is really the first CAR T therapies. And there's several of these models, several of these products that are out there. But this is really the first one that works. And we know that they're just going to get better. They're going to become more effective. They're already working on this. They're going to cause fewer side effects. And one of the things that scientists are really working on, like the other panelists on this, are beginning to predict who is it that's going to benefit from this and who's going to be at high risk for the adverse effects. And again, as a patient, I want to point out that cellular therapies such as transplants, bone marrow transplants, or allogeneic hematopoietic stem cell transplants, and CLL are the only proven curative therapy. And CAR T is a cellular therapy, and it's likely going to be a better cellular therapy. There's no graft versus host issues. There's all kinds of things that make it better already at this point, in my opinion. I told you I'd be the upbeat person. But also, we're so early in the beginning of it that it's very likely it's just going to get better and better. So I just wanted to share my personal results. And so I had no cancer in the bone marrow or blood. All my lymph nodes had shrunk back to normal. This is by a test called flow cytometry that looks down to, so these tests were by next generation sequencing and by flow cytometry. I again was restaged at 6 months, this time by flow cytometry, which goes down to 1 in 10,000 cells, and there was persistence of the undetectable, what's called minimal residual disease, MRD, undetectable, both in the blood and the bone marrow. And also what made me very happy was that I still had persistent CAR T cells by this test, polymerase chain reaction, which looks for about 1 in 100,000 cells approximately at 6 months. So my CAR T cells are still out there playing whack-a-mole with any cancer cells that might want to pop up later. This also means that I have no B cells, so I have no CAR T cells, and I also have no normal B cells. So this is just a quick picture here. That we were in Seattle, and my wife is afraid of flying, but I got her on one of these seaplanes to kind of fly high. This is a celebration. And this is how I looked really a month after the CAR T therapies. I essentially fully recovered. I was mildly anemic and stuff, but we were living life to the full, and I've been celebrating ever since. And I just want to thank everyone for this opportunity as a patient to experience this kind of scientific breakthrough. And for me, it's really been a life-saving therapy. So I'll stop there and hopefully have a little time for questions.
Yes, thank you, Dr. Kaufman. We will go ahead and take some time for questions now. Just as a reminder, please be sure to type your questions into the question box in your webinar control panel if you have not done so already. We will take a second here for some questions to come in. Okay, Dr. Kaufman, for you, can you speak more about the side effects that you experienced?
Sure. So I had so at the Hutch, it's done on an outpatient basis, and if I get certain symptoms, I'm supposed to call in. So about 10 days after I got the CAR Ts, I started to run a fever and feel achy, and so they say check the temperature again after an hour, and I did, and it was high. So my wife and I drove in, and I was admitted to the hospital for about a week. Actually, I think it was 6 days, and it was like a really bad flu. I mean, it was really achy and muscle aches and pains and sweating and temperature. And what was interesting was when I was an outpatient, I couldn't take any Tylenol, any acetaminophen, because they wanted to watch my temperature. But once I was inpatient and they were monitoring me, I could start taking medications to make myself feel better. It was awful, but it wasn't too awful. I had another complication where I was readmitted again about a week later, where I had some inflammation in my joints, and I actually, myself and some of the other docs involved in this, actually wrote up a paper on this, and we think it was related to the cytokine release, but we think I might have had a gout attack or something that was created associated with the inflammation, and that was actually much tougher. But as soon as I got the medications that reversed it, a drug called tocilizumab and IV steroids, literally within hours, I was pretty much back to normal again. So it was completely reversible. It was very unpleasant, but I got what I went for. I got the results that I went for, which is to be cleaned up of a very aggressive cancer that had failed all the standard and novel therapies. And so I certainly would go through it again in a heartbeat.
Okay, thank you. This next question, I believe it's for Dr. Comanduri. Do you encourage patients that have no other options to advocate for access to this therapy?
So absolutely. I think, advocacy from patients like Dr. Kaufman, obviously who's seen both sides of the waiting room, and from all of us as scientists, and we need to really collectively, this is something that Medicare does not move quickly. It's a phenomenal program that really is a backstop and ensures us for most conditions that we get when we're older. But these are unprecedented therapies, and we need advocacy at all levels. So this is one of those things where it really matters, and I think that there's actually bilateral support, on both sides of the aisle. This is not a partisan issue. Most people believe that both at the research level with the National Institutes of Health and with Medicare and public payers, that we need to find ways to do this, but we need to advocate for for people to kind of cut through the fog and get it done, because there are literally lives depending on this. As Dr. Locke pointed out, there are 26,000 patients a year with non-Hodgkin's lymphoma, probably 15,000 patients in the last year who meet the criteria that we would use for these therapies. Probably about 1,000 patients who have gotten them, and we don't know why there's that gulf. Is it a lack of centers right now? Is it a lack of access? Is it a lack of understanding? It's probably all of those things, and we need to really work together as advocates with patients and community members.
And a next question for you, Dr. Comanduri. Regarding your slides, how do you know if you are part of the patient subset that is sustained by CAR T therapy alone? Is that, I guess, through trial and error or a blood test? How would a patient know that?
That's a great question. Unfortunately, we don't have great biomarkers, and Dr. Kaufman, I think, said this, that for a patient who's gone through these therapies and feels that they're likely to be cured, there's always a bit of looking over your shoulder. And we don't know. There are actually patients who don't have persistence of the cells, who probably have eliminated the cancer, and even though the cancer cells aren't around, kind of keeping their foot on, the T cells aren't around, keeping their foot on the cancer cells, may not relapse again. There are other individuals who do have circulating cancer cells for a long time, but their cancer kind of outsmarts the T cells and can develop, can, for example, lose the expression of the target that the cancer cells and the T cells recognize. So, cancer is difficult. It's a little bit like a predator and prey relationship. If you have a good hunter, you can always have somebody who can kind of elude the hunter. And so it's a tricky situation. This is where there's a lot of science and a lot of understanding. I think that Dr. Kaufman's enthusiasm is correct. These are the early generations of these therapies. I think as we see more iterations of these approaches. We met with some people yesterday and are going to be, we hope by early next year, doing a trial where we target 2 antigens rather than 1. And there are a number of other approaches to deal with this. But I wish I could tell you that there was, a test that could tell somebody, are they, going to be relapsed, at some point or not. Most people will, who sustain responses, to 6 months will be long-term survivors, but unfortunately not everybody. So the relapses tend to happen early, but there isn't, what we call a great biomarker, which is how do we use tests or surrogates to predict when a good or a bad thing happen. And that's true across medicine and still something that we're learning about here.
Perhaps as a follow-up to that, can you speak to vital organ testing? Are low or high normal levels exclusionary?
To some extent, we modest abnormalities in liver function or even heart function or kidney function can lead, to therapies like this being given. But because of the risk, for example, if you have that inflammation like a cytokine release syndrome, which is a bit like a severe flu or even a bacterial infection, if you have severe kidney function problems going in, for example, you're much more likely to have a fatal complication. Now, I think that Dr. Locke and myself and others are used to taking care of really sick patients and wanting to give them the benefit of the doubt. And the more desperate their medical situation is, the more likely we are to kind of tolerate, a patient who's not looking great. And often patients are reduced to a poor functional status or have these organ function problems because of their illness. And if we think that that's the case, we might go ahead and treat anyway. So I think the important thing, and I think Dr. Locke would agree with me and Dr. Kaufman would agree with me, is seek early evaluation at an academic medical center. No academic medical center worth its salt, like Moffitt or Sylvester or the Hutch, where Dr. Kaufman was treated, is ever going to use a therapy too early or in an inappropriate situation. If we think somebody's likely to be cured without what we're doing, we're going to say, there's nothing else that you need to do. So seek early evaluation, and because sometimes what happens is that toxicities progress because people don't refer early enough, and we lose our window of opportunity. So I think that's something that we will describe where your risks are going to be higher if you have certain issues, and we will try and find a way to get patients to therapies that are curative as long as we think it's safe.
Great, thank you. Last question to Dr. Kaufman. Any clue as to when Medicare may make any kind of decision on approving greater payment for this therapy?
That's another great question, and I have no sense. I mean, when we met with CMS, we met with patients. It's a room full of epidemiologists, essentially statisticians trying to figure out, looking at the costs and the benefit ratios, which is what they should do. And we were trying to put kind of a human face to that. I don't know, maybe some of the other panelists would like to address that and might have a better sense. I didn't get a sense that anything was imminent. That was clearly my sense.
Yeah, I think that there are a lot of partial fixes. For example, this new technology add-on payment was greater than historically has been there, but the payment methodologies, we literally submitted a 22-page letter, we meaning the ASBMT, and a week ago actually, and then yesterday, and this is a sign of at least progress. The ASBMT and the American Society of Hematology were asked to actually brief Senate staffers on this. And I'm actually talking at an event in Washington at the National Press Club, sponsored by this MIT group that's trying to find financing models, and there will actually be senators on both sides of the aisle. So we have to get out there, and I would say for anybody who's interested in this stuff or has a family member or relative, or even if they just think, hey, if I should ever have to have this, I want access, right? This is one of those things where sending the emails, picking up the phone really matters. So just advocacy is important at all levels, and this is really, we need participatory democracy, and we want, we certainly live in a great country with respect to healthcare, and we want to be able to provide these to everybody who needs it, at the right, in the right indication.
Great. Well, we are at time. Thank you, presenters. Thank you, audience. We appreciate your time and engagement. If you have further questions about CAR T cell therapy, depending on their nature, please direct them to your personal physicians or today's sponsors, Glasgow, Gilda's Club of South Florida, and the CLL Society. Thanks again for joining us today.